To Ahmad coaching, and this is your teacher Dr. Anam. Today we are going to study about enzymes. Enzymes are protein that increases the biochemical reaction and not consumed at the end of the chemical reaction. Let's take some examples from the daily life. For example, you had a breakfast at about 9 o'clock, had a heavy breakfast, but again at 1 o'clock you are hungry. What happened to the food that you had as a breakfast at 9 o'clock? Why you are hungry at 1 o'clock? Yeah, needs to think about that. Okay, similarly, you have a shirt, and on the shirt, when you are eating something, some sports or some kind of food is dropped. Now what you do, you use detergent to wash it, and after that you have a new clean shirt. So where did all these dirt, all these kind of particles that were on the shirt goes? So this is another mystery, but this is no more a mystery, because the answer to these questions is enzymes. Enzymes present in your stomach digest your food that you had on breakfast. That's why you are hungry at 1 o'clock. The enzymes in the detergents, they remove the dirt particles on your shirt, and after that you have a new clean shirt. So in other words, enzymes are very important in our life, not only in our daily life but also in our body. So let's talk about more in details about the enzymes. Like I said before, that enzymes are actually proteins. The proteins, they are made up of amino acids. What happened, that amino acids in the proteins, they join with each other in a very specific sequence. This sequence in every enzyme is different. For example, if we take example of enzyme 1, this sequence might be 1, 2, 3, 4, 5. And if we take example of enzyme 2, this amino acid sequence might be 5, 3, 4, 2, 1. In other words, every enzyme has a specific sequence of amino acids. Because of this specific amino acid, the shape of enzyme is different from each other. What happens, that these amino acids, they join with each other and then they fold on each other like this and form a specific globular shape. Here you can see this is a globular shape. These amino acids, they are folded or coiled on each other to form this shape. So the enzyme has a globular shape, and this globular shape is because of the arrangement of amino acids. This arrangement in enzyme 1 will be different, and the same arrangement in enzyme 2 will be different. For example, this is enzyme 1 arrangement. The enzyme 2 may arrange in a different way. So amino acid sequence is different. Because of that, the globular shape of each enzyme is different, and this is the reason that each enzyme is performing a specific function. The shape, if we talk about the shape of enzyme, the enzyme has a globular shape. For example, 100 amino acids, they are involved in the construction of this shape. About 3 to 4 amino acids, they will make a specific region on enzymes, which is known as active site. This active site is very important because this site is related to the catalysis function of enzyme. What happened? That enzymes, when they are going to perform its function at the active site, a substance is going to combine like this. This substance is known as substrate. So any substance that is going to attach itself with the enzyme or on which an enzyme is going to act is known as substrate. After the attachment of substrate with the enzyme at the active site, this substrate is broken down into products. These are the products. And this is how enzyme catalyzed reaction takes place. What about other amino acids? We said only 3 to 4 amino acids they are present at this active site. The rest of the amino acids, they are used to maintain the globular shape of the enzyme. This globular shape of enzyme is very important. Any change in this globular shape may result in the denaturation or the destruction of enzyme, and if the enzyme is destroyed, this enzyme cannot perform its function. When we talk about the enzymes, some terms they are used. The first term is cofactor. Cofactor is any known protein part we know that enzymes are protein. So if any non-protein part is attached with the enzyme, that part is known as cofactor. For example, we have an enzyme, and with this enzyme, any non-protein part is attached. This non-protein part is known as cofactor. The cofactor act as a bridge between enzyme and substrate. If the cofactor is present at the active site, what happens that this cofactor will help in the attachment of substrate with that of the enzyme. In other words, it is acting like a bridge. And this is very important for enzyme to perform its catalysis function. Along with this, the cofactor, it also provides chemical energy. This chemical energy is very important to perform catalysis function. If we talk about the cofactor can be inorganic. It can also be organic. The inorganic can be metal ions, for example, magnesium, iron, copper, or it can be organic, which is vitamins. In the context of cofactor, different other terms are used. First is activator. Any cofactor which is detachable from the enzyme is known as activator. It means that it attaches itself with the enzyme, but after its function is performed, it will remove itself from the enzyme. Along with this, we have prosthetic group. Any cofactor which is attached with the enzyme by covalent bonding is known as prosthetic group. For example, we have an enzyme and we have this cofactor, and then covalent bonding develops between these two, then this cofactor is known as prosthetic group. Along with prosthetic group, coenzyme word is also used. Coenzymes, they are also attached with the enzymes, but their attachment is very loose. So if they are loosely attached with the enzyme, they are known as coenzymes. Most of the coenzymes, they are organic in nature. For example, vitamins. They perform very important function inside the body. And another property of coenzyme is that just like the enzymes, they can be used again. It means that at the end of the reaction, they are not utilized or not consumed, but they can also be used again and again. Another term used is known as Apo Enzyme. So if you have an enzyme without any cofactor or coenzyme, it is known as Apoenzyme. And another term is holoenzyme. In holoenzyme, cofactor or coenzymes, they are present. It means in this example, this is a holoenzyme. Why? Because here we have cofactors, right? In this example, we do not have any cofactor here, so this enzyme is an apoenzyme. The cofactors, they are very important because they activate enzymes, and if the enzyme is activated, it can perform its chemical reaction, or it may speed up a chemical reaction. Now comes towards the location of enzymes. The enzymes, they are normally present in the cytoplasm. Here we have a cell, have nucleus. This part here is cytoplasm. So inside the cytoplasm, you can find enzymes freely floating. Along with this, the enzymes, they can also be present attached with the subcellular organelles. These organelles, it may be mitochondria, it may be nucleus, the nucleolus, or it may be any other like ribosomes. Or the enzymes, they can also attach itself with the cell membrane. If we talk about the importance of enzymes, the enzymes, they are very important because they are involved in metabolic reactions or metabolic processes such as respiration, photosynthesis. All these reactions, they need enzymes. So if the enzyme is present in a cell, these reactions, they can take place. So this tells us that how much important these enzymes are, and in other words, life is impossible if enzymes are not present. So that was all about enzyme, its function, and some terminologies attached or used with enzymes. If you have any question, you can ask me in the comment section. If you like this video, please share it, like it, and subscribe to my channel. I will see you in the next video.